is the lead in to the first computer practical, which is about database querying and exploring homology alignments with a bit of software called BLAST. And we'll look at that now. So again, these are the aims for today. It's to understand the different types of biological database that's out there and to search these using both sort of text queries and also sort of nucleotide and protein sequence similarity using something called the BLAST tool, which stands for the Basic Local Alignment Search Tool. It's also important that you're able to critically evaluate any output from a BLAST search. So essentially, what is a database? And hopefully you're kind of aware of this already. You've searched databases all your life and you continue to do so. So every time you type something into Google, for example, or iTunes or Spotify, you are inputting what we call a query, and you're retrieving information that matches that query from a collection of data, which is searchable, updated periodically, and generally cross-referenced as well. And that is essentially what the database, or what a database is. So it's searchable in that there's often an index and kind of some form of content page, and you're able to search through that list. It's updated sort of periodically. Actually, in biological databases, this tends to be sort of actually every day, and you have kind of different release versions, and you can often find that information when you search the latest database. It's cross-referenced as well, so there are often hyperlinks on said databases to link to other databases out there. So you may have a nucleotide sequence that you're searching, and it may have a link to a protein database where you can get kind of 3D and structural information from that other database. It's worth noting that different databases contain different types of information. So there's a database called the NCBI, or the National Centre for Biotechnology Information, and this is a common sort of database that we're actually going to use, and this contains a lot of information, and it actually sort of links to various other databases as well, so it's a good one that we're going to use. But you also have specific databases as well. So the Protein Families database actually contains information about protein domains and kind of protein information. And also different databases serve different communities and purposes. So something like Wormbase, for example, or Flybase, they serve specific kind of organisms. So the Flybase, for example, is specific for Drosophila melanogaster, which is a fruit fly. It's a kind of model organism. And again, Wormbase, sort of C. elegans is the organism on there. And those databases are very specific for those organisms, but the NCBI is pretty generic, actually. There are also tools for recording data in various formats. So you can kind of retrieve your data in various types of kind of output for various different programs and downstream analyses, which we will get to later on. Today, and for your computer practical, we're pretty much going to just limit our searches to nucleotides, so nucleic acids, and protein databases. But there are many others out there. It's worth saying that not all databases are indeed equal, and they vary depending on things like the type of data. So you have, for example, nucleotide databases versus protein databases. In our case, NCBI has both, but you can have specialist ones. The breadth as well. So again, I mentioned this. You can kind of have sort of all organisms represented in a database or specific. So as I mentioned, Flybase is an example with the fruit fly. You won't get any human biological information from Flybase. It's also the amount of data that is kind of, or that varies between different databases. And we have these two terms, redundant and non-redundant. essentially redundant databases have multiple inputs of a particular gene or transcript or protein that's essentially represented many, many times within the database. A non-redundant database, as it says here, there is only one example of any given sequence, and for NCBI it's designated as NR, so non-redundant. I'll talk more about that in a minute. There's also a difference between redundant and non-redundant databases in the terms of the quality of the data that's produced. So redundant databases, where you have more than one representation of any given sequence, often tends to be automated. And what we mean by automated is that it's kind of a computerized system that has dealt with that information, putting it onto and storing it on the database. Non-redundant databases are generally non-redundant and only have one example of a given sequence because they're manually curated. Not always, but it's a kind of generalization there. And essentially manually curated databases have been gone through with a fine-tooth comb by scientists, by us, to ensure that sequences, for example, are not repeated, that they're of high quality, and that they're correct. So, an example, this is the translated EMBL database, and it's automated annotation. So this database has lots of sequences because it's done by a computer, so the kind of processing power is greater, so it contains a lot of sequences, and it's obviously redundant, but the sequence quality of those sequences is actually pretty low because there has not been manual annotation or curation by sort of humans, essentially. SwissProt, again, another database. You're not going to use either of these, but it's just worth bearing in mind that actually this is the opposite. So it has far fewer sequences. Okay, it's non-redundant. So non-redundant databases tend to be smaller, but the sequence quality of those sequences of the information stored is much higher. As I said, we are in the computer practicals going to be using the NCBI reference sequence database, which is abbreviated as RefSeq. It's non-redundant and it's very well annotated, and it includes both protein and nucleotide sequences. So we are generally speaking going to be using this. It's a pretty good, solid database actually. So when you log on to the NCBI homepage, it looks something like this. The website link is down here, but it's also in the course handbook and on Teams. But you can just type NCBI into Google and it will bring up this page. So it's worth noting a few things about this page. On the right-hand side here, you have popular resources. So again, these are links to kind of popular sites or parts of the site that you might need to use. So PubMed, for example, is a kind of database for medical literature. So if you're looking at kind of searching the scientific literature, then that's a good place to go. BLAST, so I mentioned the basic local alignment search tool earlier on. So that's here. So not only can you type it into Google, but you can actually just click here. If you want to search a nucleotide database, again, there's a kind of link there. And a protein database as well. So they're kind of the ones to watch out for. It's worth noting though that you can actually just click this arrow up here and you'll get a drop-down list of every single database that you can possibly search through this homepage. So if you wanted to search, for example, the protein database, you can just select protein. What else is there? There's the nucleotide database. There's OMIM, so the Online Mendelian Inheritance of Man database. So that's good for searching human genetic diseases, which we will, again, you'll have a go at. And there's various other ones as well. But those are the three that you're going to be using. Okay. Again, there are kind of links here. I'm not going to cover this because actually it doesn't matter too much to you, so we'll just move on. When you're searching, it's important to be aware of how you can refine your search to search more intelligently and not kind of return thousands and thousands of hits when you're only looking for something rather specific. And you can do this through the use of what we call Boolean operators. So I'll just take you through that now. If you were to type the word and, so for example, 1 and 2, what's returned from the database must match both 1 and 2. So if you were looking for Homo sapiens, for example, if you typed Homo and sapiens, it would bring back all the results for both of those terms. Or means that it will match up one or the other, essentially. So it will either match Homo or it will match sapiens, and it will bring back those results. So it doesn't have to match both; it can just have either or in the title. Again, you can use not, so it must not match. So you can bring a query into the search engine, and if you put not and then type something else, it will bring back everything that does not include whatever it is you type after the word not. It's worth noting that the default behavior is and. So if you don't type anything in between words and you just put a space bar, the database will immediately treat that as an and function. So just be wary of that. Terms are processed from left to right, obviously, but you can change this by using brackets. So, for example, if you were going to search CDK or and then brackets cyclin and kinase, it will first of all search what's in the bracket. So it will make a search for cyclin and kinase. So again, the sort of union of both. So it'll have to match both cyclin and kinase, not or. So it will match both of those, and from that output, it will then try and match CDK. Okay, so it'll either match that or CDK, but it will do a search for cyclin and kinase first. That's kind of reversed over here. So if you were to put brackets over CDK or cyclin, it will do this initial search first, and from those results, it will then try and match with kinase as well. You can use quotation marks to force text as one phrase. So if you don't want an and to be kind of immediately, as I put here, put in between two words, you can just put quotation marks around them. So if you didn't do that and you just put public health, it would instinctively, the database would put an and in between the two. If you were to put them in quotation marks, that wouldn't be the case. So it won't search for public and health. It will just search for public health as kind of one phrase. It's important to note that there's a wildcard as well, which can be used with the star symbol. So if you put this after a particular point, anything downstream of that will be brought up by the search field. So if you put trypanosome here, it will bring back anything with either an E on the end of that, an A, or soma, or trypanosomatids here. Essentially, it's a wildcard, so it will allow you to search for variants of that particular word. Finally, you can narrow things down even more by specifying the fields within which to search for your particular query. So, for example, Homo sapiens or kinase, you can actually specify that you want Homo sapiens to be found within the organism field. So just because Homo sapiens is mentioned in like the title, for example, that won't return the search because it will only search for it within the organism field. And again, kinase here, again, if that's in the title of a paper or something, or a transcript, then that won't necessarily come out of the search, because you're specifying that it needs to be contained within the gene name of that particular search. More details on that later on. It's important to note that sometimes and very often you will get far too many results, or indeed too few in some cases, from your query. So really it's just trial and error. I would say you try and refine your search terms, so have another go essentially. Try and use those Boolean operators and maybe a wildcard, things like that if you can. Refine the search using those square brackets into fields, and just try again essentially. And there's not much else you can do. Just try and refine that search query, and you'll get better at it over time. It's just worth knowing that the advanced search feature is actually linked on the or near to the search bar, and will allow you to combine past queries using the history feature. And you can also limit your fields during that sort of process as well, so you don't have to type the square brackets into your query. You can search afterwards and limit your search to restrict to, for example, specific organisms, etc. So just to show you that, here we've typed to do with Huntington and Homo sapiens, and if you look here, there's the advanced button, and if you were to click on that, you can restrict via field. So if you click that drop down menu, you can see all the different fields. So as we said, organism, title, gene name, etc. And you can just restrict what you want to be searched in this bar here, and you can just add more. It will also tell you what you've searched for, so you can go back to the history function here, and you can see what you've searched for previously. So, how can we search? Well, we can search sort of using text, so we can type the gene or the protein name. As mentioned, it can be kind of ambiguous in some cases. Or you can search via an accession number. So if you know or have access to this accession number, which is often cited in the literature, you can just type that in and it will take you immediately to that sort of gene or protein. But you have to know these. So if you've just sequenced a bit of DNA yourself, you can't actually do that because you don't necessarily know what gene it is, you don't know what protein it is, and you won't have an accession number for it. So what can you do? Well, you can search, as alluded to in the first lecture, you can search by sequence similarity rather than any specific name. So what essentially this is it's taking your, let's say, protein sequence. You can use a blast search to search essentially for similar sequences in the database, and it will bring up a list of similar sequences, and it will tell you information about how similar those sequences are or not to each other. So you can then make an informed judgment as to whether what you're looking at is the same or related to the protein that's returned from that search. So how does this blast search work? Well, you input your query, which is designated by this red line here, into the database, and you click search. What it will do is it will actually chop up your query sequence into loads of little kind of seed sequences or kind of, bite-sized chunks essentially. And what it will do is it will search for identical matches to each of those little sort of seed sequences from or to sequences that are already present in the database. So those sequences are represented by the black line. So these are the sequences that are within our database, and it will search for identical parts or chunks, and it will line them up with each little bit of seed sequence. Once it's done that, it will then try and extend the match. So let's say we've matched this little bit here. So we've matched these three, let's say it's a protein, it's 3 amino acid residues. What the system will then do is it will try and extend locally that alignment and look for identical matches only. And it will do that with every single bit of seed sequence that it's found. And then it will give a score for how good that match is or not. And that will depend on how identical the residues are and if any gaps have to be introduced. These results will then be shown in the BLAST output for you to look at, which we'll have a look in a minute. So how do you perform a BLAST search? Well, you can go to the NCBI database and you can select BLAST over here on the right hand side. It's probably the easiest way to do it. Once you've done that, you end up with this page, and there are various different types of BLAST search that you can select. Now, to give you a bit of a hint for the computer practicals, we're going to either be using nucleotide blasts or protein blasts. The other two we're not, but it's just worth sort of letting you know what they are. So, if your query is a protein sequence and you want to search a database of protein sequences, so we're comparing proteins with proteins, that's called BLAST-P or protein blast, which is this one here. The nucleotide blast up here is not actually represented in this table, but essentially if you're using a nucleotide sequence and you want to blast a nucleotide sequence, then that's just the nucleotide blast here. The other two, which are kind of not actually going to be used by you, is if you, for example, have, let's say, a translated nucleotide sequence and you want to compare to a protein database, it's called BLAST-X. If you have that translated nucleotide sequence and you want to compare it to a transcript database of other translated nucleotides, then that's tBLAST-X. And again, if you have a protein sequence that you want to compare against a translated nucleotide or transcript database, then that's tBLAST-N. But don't worry too much about those. The main one really for you is BLAST-P or protein blast. So what happens? Let's pretend we're doing a protein blast. You end up with this particular window once you've clicked that, and we'll just go through this now. So you place your query sequence into here. It has to be in the right format, so it has to be in what we call a FASTA format. Don't worry too much about that. Your computer lab practical, I've kind of written instructions as to how you obtain your query in FASTA format. It's pretty easy. It has to be in the right format for the system to read it. You can insert a title here if you want to, and all the way down here you can see you can select the database that you wish to search. So here, well, I've gone for the non-redundant protein sequences database, just NR. If you wanted to search RefSec, that's also down there as well. So you'd click this button here, and that would allow you to select for RefSec. You can, if you want to, search for a particular organism. So if you know that your protein sequence comes from E. coli and you therefore want to search E. coli, you can type that in here. Or if you want to, take an E. coli sequence and search and see if there's a homolog in humans, then you can search a human protein database by typing humans into there. And it will automatically fill once you start typing. There's various other bits sort of downstream of this, but I'm not going to worry about that too much now. So the organism can be placed into there. Once you have then submitted your search, so I here have submitted a protein sequence from a HEL308 helicase from Archaea, so Halopharix vulcanii, which is an organism I work on in the lab. You get some information here, so it's told you what it is. So it's an unnamed protein product because I've not told it what it is. I've searched the non-redundant database using BLAST-P, and it tells me how many amino acids my query length is. It's 827. You'll see down here that it actually brings up the results in a kind of sort of tabulated view, and you can see the top hit here is an ATP dependent DNA helicase from Halopharix vulcanii, and it will give you a score. It will give you query cover, and we'll talk more about those in a minute. Essentially, it's how much coverage that match kind of covers from the sequences within the database. Something called an E-value, which we'll get to, and it will give you a percentage identity, so how many residues are essentially identical to that in the database. So this particular sequence here has been brought from the database. It's 100% identical to the sequence that I've inputted here. So again, search summary is up there in the top left. We have view tabs here, so you can click on graphic summary, which I'll show you in a minute, which actually allows you to see this tabulated view as a kind of graphic, so you can see whereabouts those alignments have occurred. And also the list of hits is here. So again, it will give you many, many hits that have been found, but the top hit is usually the top of the list of hits. So if you click graphic summary, it will actually show you the hits in red here and pink and green in this case, and it will line it up to your query sequence here. Up here, it will actually give you any kind of protein domains that are found, but don't worry too much about that for now. We're more interested in just the sort of coverage of the hits in the database. You'll notice up here that the colours actually link to scores of alignments. So red here is significant alignments. So this is just a score. And actually, you can see this top hit here is absolutely identical for the entire length of the query sequence. So that was the top hit that we saw last time, or in the previous slide. Just to go back, if you were to then click on this particular alignment here, the ATP-dependent helicase, or you go to the alignments here, if I do that, you can see the actual alignment that's been made. So this is this was not the top hit, but it was because the top hit was identical, but this is just to show you. You can see that the query sequence here is at the top. So it's the top line of each kind of segment here is the query sequence that I inputted into the database. And you can see here the subject is essentially the sequence from the database that it's brought out. And in the middle here, it will give you information as to whether that sort of residue is the same or whether it's similar. But don't worry too much. I'll talk about that in a minute. So let's zoom in to that particular bit, and we'll just talk about some of the things here. So you have a bit of information here. First one is identities. So essentially, this means 272 residues here have been matched as identical. So they absolutely match 100% the query sequence, but it only makes up 50% or so of the residues. The positives include those that are identical and any that are similar. So as you hopefully recall, not all amino acid changes are that different from each other. And so this will include any residues that are different but perhaps very similar, which are actually designated via a plus here. So for example, leucine here and valine are actually pretty similar in terms of their chemistry. So there's a plus that's been put here. Gaps are also included, so there's 6% gaps. So that's where there's not been a match that's been able to be made, and they're determined by these little arrow hyphens here. And essentially gaps are required to be placed into the alignment to ensure that the alignment is correct. Because it might be that, excuse me, some of those amino acids are either not present in your query, or there are amino acids that are present in your query that aren't in the database. And they need to be included, but obviously it would throw the alignment out for the rest of the amino acids. So they're designated by a gap. The other thing just to point out here is it's given a score. So the alignment's given a score. Generally, the higher the score, the better. The important thing here is the expect value, or the E value. This is a number that is essentially the number of hits you would expect to see by chance with the observed score or higher, given a database of the size that you are searching. So just let that sink in. It's a bit of a weird one. Essentially, if you have a low E value, so here we've got 2 times 10 to the minus 137. If you have a low E value, then that is a lower likelihood that the hit is shown, or that the hit shown has this score or higher due to chance, essentially. So the lower the e-value, the more likely it is that this is a real hit and not just something that is, come out of the database search by chance because the database is so huge, essentially. So we're looking for a low e-value here. The lower the e-value, the better. Generally, there is no cutoff for the e-value. Academics tend to work at about 1 times 10 to the minus 6 as a good estimate for whether things are kind of a sort of real match or not. So this is clearly far lower, far smaller than that. So just as an example for e-values, if we were to look at this top hit up here, the e-value is 1 times 10 to the minus 40. So we would say that's a pretty low e-value, and there's therefore a low probability that this alignment has occurred by chance. Okay, in other words, there's a low probability that it's just a random match and that there's no relationship between the two sequences. All the way down here, we can see that this e-value is much higher, so it's 3.2. So there's actually quite a high probability that this is just a random match and actually means nothing. And you can see that this is related to the score because actually the higher the score, the lower the e-value, as you can see here. So again, the lower the score here, the higher the e-value. It's worth just pointing out, though, that e-values can go wrong. So this is a protein sequence I found from trypanosomes. And what you can see is that actually the problem with trying to match this sequence is that actually there are a lot of repeating units of these amino acids. And actually, I've highlighted them in red. There are loads of them. So the database searching and the e-value hits all depend on the assumption that your database is made up of random sequences. But proteins are not made up of random sequences, as you can see here. These particular motifs, these kind of sets of amino acids, are repeated many, many times throughout the sequence. Okay, so it's not a random sequence at all. It's repeated for reasons in terms of biochemistry and kind of how reactions work. But it's repeated, and therefore that's going to kind of affect your e-value score. So real proteins like this have many, many repeats. Again, this is another example of where e-values can go wrong. The expect value here is pretty low, actually. So it's a low probability that this could have occurred by chance. But you need to use your brain, because if you actually look at the match, look at the sequence alignment, you can see that there are lots of proline and lysine residues within this sequence. So proline being P and lysine being K, there are lots of them repeated. Okay, so actually it's matching a very, very low e-value, but it's essentially probably more likely because there's just lots of repeats of those particular residues. Okay, those proteins may not necessarily be homologous to each other or related to each other. There are lots of these just repeated units, similar to this example, that mean that the e-value is low, but actually it could well have occurred by chance because of the repeating units. So you just have to use your brain to then decide whether you think that this is a real match and how much those repeated amino acid residues actually impact your thinking as to whether this is a real match or not. So as I said, there's no real cutoff. Use the e-value to inform your decision making, but don't use it as a kind of binary cutoff value to tell you whether something is indeed a homolog or not. So the final thing I just want to talk about is homologs. There are different flavors of homologs. So homologs, don't forget, it just means inheritance from a common ancestor. So everything here is a common ancestor to this particular common ancestor down here. Therefore, everything is homologous to each other. But we have sub flavors, if you like, of homologs. So we've got orthologs, which derive from a speciation event, and paralogs, which derive from a gene duplication event. So just to sort of work through this, let's say we have an early gene of interest here that duplicates into an alpha chain and a beta chain. So it's just a gene duplication event. Because this gene duplication event has occurred, this can then kind of be passed through to different organisms. So let's say that the alpha chain, for example, here, sort of this ancestral organism then sort of diverges into a frog, chicken, and mouse. These are all orthologs of each other because this alpha chain has speciated essentially. It's through speciation has been incorporated into a frog, a chicken, a mouse. But because they've all derived from a speciation event of this original organism, these alpha chain genes within the frog, the chicken, the mouse are actually orthologs or orthologous to each other. And that's shown here with the beta chain as well. But paralogs derive from duplication. So the gene duplication event has arisen here. And if we were to look at the mouse beta chain and the mouse alpha chain, they have arisen as a result of the original gene duplication event up here, and therefore they are paralogs to each other, as are, for example, the chick beta chain and the chick alpha chain, and the frog beta chain and the frog alpha chain. So essentially, it's all type of homology. It's all descent from a common ancestor, but it's just worth bearing in mind that orthologous sequences and paralogs also exist as kind of sub sections or sub flavors of homology. If you're confused by that, do speak to me. And it's a bit of a abstract concept, but if you just work through that, hopefully that will make sense. But again, do speak to me or email me if you are not sure. This is really important actually, and kind of one of the final things I'm going to say, which is that the output of a BLAST search with an e-value and showing you the alignment and giving you the scores tells you about how similar those sequences are to each other. It does not tell you if they are homologous or not. But the key thing is you can use the degree of similarity and the e-value, and obviously looking at the sequences yourself, you can use all of that information as an argument to say whether those sequences are either homologous to each other or not. So the BLAST search does not tell you about homology. It just tells you how similar those two sequences are. Okay, what that allows you to do using your brains and the output you get is it allows you to then make an inference as to whether they potentially are homologous or not. That's a really important point. So BLAST is a tool. It's not a kind of, it's not like a binary threshold where it tells you, yes, that's homologous, no, it isn't. Okay, you have to use your brain to decide, using all the information that you are given, whether you think it's a homologous sequence or not. Or indeed orthologous or a paralog as well. So the summary's here. Hopefully you can do all these things. Essentially, the most important thing is you can read through all this, but don't be a naive user. So use these things, but think about the output that you're getting from these BLAST searches. Don't just kind of click buttons and not switch your brain on. Okay, you need to actually engage with the process and think about what the information coming back from the BLAST search tells you about the sequences that you've put in. So keep your brain switched on. That's the end of this kind of podcast, but the computer practical will be the following session after this. You'll be provided with the instructions. I'll print them off for you so you can write on the paper and kind of work things out. Essentially, bring your surface, open up the websites listed in the instructions, the links to which are on the team site and in the handbook, and then you just work through the exercises within the Word document that I'll give you. It will give you a step-by-step set of instructions as to what to do and where to go and what to type in, etc. And every so often there'll be a question relating to the search or the blast search or something that you've just done, and then you can just hopefully answer that question. I'll be there during the session, so do ask questions, or try and let me know of any issues that arise. You may or may not finish all questions within the 40 minutes that we have, or 35 minutes that we have. So if you want to continue doing that quiz, you can afterwards, and I'll provide the answers at some point after the computer practical has kind of officially ended. If needed, I can record a podcast going through exactly how you get to each answer, but I'll only do that once kind of everybody's had chance to have a go at the quiz. But you feel free to email me or speak to me during the session.